Kiyun is the first one, mechanical engineering PhD student for about 3 years now, I suppose. Kiyun does both modeling and experiments. Kiyun, the floor is yours.
You turn on both. I can find it. Sorry. I should.
Yeah, it's working. Why is that showing things that, or it's not on my end? Oh, okay. Yeah. Yeah, so hi, my name is Ki. I'm a grad student working with Professor Tahir Saif, and today's talk is about learning and memory under mechanical tension in vitro. So one of key findings by Eric Kendall was actually plasticity at the synaptic level between two neurons. So here's an image of two motor neurons forming a connection with one sensory neuron here. And by providing a stimulus, in this case serotonin, he found that the amplitude of this the potential voltage at the synaptic level increases after 10 minutes. And by increasing the number of stimuli, he saw that the increase in amplitude in membrane potential actually remains high even after 24 hours. So what he found was this short-term and long-term facilitation is actually due to by some plasticity happening at the synaptic level. For example, more vesicles are released like this, or new synapses are formed like this one. So the objective of this study is to see does in vitro neural network have plasticity or not, because it's quite hard to measure this plasticity at the neural network since there are so many connections. It's very difficult to identify each individual connection. Then, so my main focus on this talk will be about this question. And the second question we have is, if neural network has plasticity, then can we tune it? In our lab's perspective, we are interested in tuning this by mechanical tension. As we all know, neurons communicate with each other chemically through vesicles and neurotransmitters. However, the many study shows that this mechanical tension actually contribute to have a significant role in neural communication. Here's a schematic of showing one way of how mechanical tension contributes. Here I have a presynaptic neuron and with synaptic vesicle at the terminal and postsynaptic neuron with receptors and ion channel. And when you pull the axon of presynaptic neuron, and then more vesicles are accumulated at the synaptic terminal. And this result, this study shows that result, you have a fruit fly, and when you pull the axon of neuron of fruit fly, then what they found was more vesicle accumulation at the synaptic terminal, so it increases. And also mechanical tension induce increase in neural firing as well. So here is another study showing this brain slice, mouse brain slice, and as you stretch this mouse brain slice using this device, the neural excitability increases. So going back to the question that we have, first, does a neural network in vitro have a plasticity or not? And if it has, then can it learn, remember, and compute? And the last question we'd like to answer is, then can we tune or modulate this cognitive functions, especially by mechanical tension? So here's the experiment setup. We use optogenic hippocampal neurons dissected from transgenic mice, cultured on the 60 channels MEA, and let them form a network. And the MEA has about 200 micron inter-electrode spacing. And we use optical stimulation for training and recall phase. Training was done either 5 or 10 minutes, and recall was done 2 minutes. And this is how the pattern looked like. And this is the raster plot showing with optical stimulation. The red box represents the duration and position of the stimulation. So in this case, light or optical stimulation was applied to every ear on MEA. And you can see neurons fire and burst following this light pattern. And then I'd like to talk about the definition of burst and network burst we use in this study. Typically, burst is defined as consecutive spikes consisting of X number of spike and above with interspike interval less than certain number of milliseconds. However, we want to define or we want to get bursts that are relevant to memory-related activities. So there is something called ripple oscillation, which is very high frequency wave in the brain above 80 hertz. So we define a burst when instantaneous burst spike rate is above 80 hertz. That means it filters out any other bursts that are less than 80 hertz. So once we define a burst, then we define a network burst, which is just synchronous bursts. And this is what actual experiment look like. So here's the layout of the electrodes again. And during training phase, we shine light both blue and red regions simultaneously. So after training, now information is embedded into this network. And then during recall phase, we provide a Q stimulus, in this case, just shining only this red region, which is you're giving a partial information, and we measure what neurons are doing or activity of neurons in this blue region. This is the just 10-second window of the raster plot. This is before any training. You can see some spontaneous firing, random spikes, and also some burst here for each electrode. And they are synchronously bursting, forming a network burst. This is during training. The spikes or bursts from under this red box are coming from electrode in this red region. And spikes from this blue box are coming from this region, this blue region. And spikes coming from these outside of red and blue are coming from here. And this part is coming from here. So as you can see, during training, of course, under light, this neuron actually fires and bursts quite well. And here's the recall phase, meaning we shine only light only on red region. And of course, neuron under this red region, they of course fire and burst. And what we saw is not always, but most case that neuron under this blue region also fire and burst, as you can see. Here's the control case where no training occurred. No training, we just cue them. And of course, neuron under this red region, they fire and burst. However, neuron under this blue region, they don't burst. They just show spontaneous firing. So from here, we found the evidence that this in vitro neural network has plasticity, so that after training, it shows different firing or bursting pattern. Now, we try to quantify this memory recall activity using synchron index, which is a measure of how neurons in blue and red region synchronously bursting. And you can get it from this equation. So we have a burst train from each electrode, and we vectorize this burst train across network bursts. So now we have a vector of burst train for each electrodes. This X and Y represent a vector of burst train from one electrode from blue and the other electrode from red region. And you can get synchron index. This is essentially equivalent to just dot product of two vectors. And this is the synchron index over time, depending on duration of the training. So no training case, 0 minute, the blue one, at the initially, they seem to have, the blue and red region seem to have high synchron index, about 0.7. And when you cue them, meaning when you shining only red region, you're in kind of intentionally make neurons on the red region fire. So you're kind of breaking the synchrony. So that's why the synchron index goes down. And for 5-minute training case, of course, at the initial point here, you make neurons under blue and red region intentionally make them fire. So synchron index is very high here. And upon cue, you can see the synchron index remain high but it decays over time, possibly indicating forgetting. In 10-minute case, the synchron index remains higher than 5 minutes, suggesting that the duration of training matters. So now we saw the in vitro neural network has plasticity and demonstrated this memory-like behavior dynamics. From here now, we want to see how tension or mechanical tension actually modulate this network behavior. So we use chemical called blebbistatin, which is a myosin 2 inhibitor, which is known drug to reduce contractility of neurons, as you can see in this figure, and also suppress neural firing. Here's the longer window of a raster plot. The green box represent the global network burst. And I administered the blebbistatin 0.1 micromole here. And clearly you can see that in terms of global network burst or network behavior, doesn't seem to change before and after the administration of the blebbistatin. And I increased by 0.4 micromole. And as you can see, when you add, there are so many instantaneous firing or spontaneous firings, random spikes. And then after 2 to 3 minutes later, this global network burst or network behavior reappears. And for analysis, I'm going to talk about later how we measure this time from here to here as a recovery time to measure the effect of the dosage amount of blebbistatin. So the reason why there are so many this spontaneous spikes rather than network behavior is we think since blebbistatin interrupt or disturb the actin myosin movement at the synaptic terminal, so whatever neuron has vesicles and it just release every vesicle that it has due to blebbistatin. So that's why we see a lot of spontaneous spikes, but no network behavior. And this is a higher concentration, 1 micromole. And as you can see, it eradicates all the network behavior, and this network actually never recovered. And we measure the network burst rate over time, depending on dosage of the blebbistatin. This line is when I inject the blebbistatin. And you can see when you add 0.4 micromole, the rate decreased significantly and it recovers, but not fully. However, when you increase the concentration by 0.1 micromole or even 0.2 micromole from here to here, the network burst rate doesn't seem to change. However, when you again increase by 0.5 micromole, it drops significantly and then it recovers. And I measure this recovery time versus the change in concentration. And as you can see, at the lower concentration, 0 or 0.1, 0.2 micromole, there's nothing to recover. So this recovery time is quite close to 0. And about 0.3 or 0.4 micromole, it takes about 2 to 3 minutes for network to recover fully. And above 0.6 micromole, the network never recovers. So from here, our future plan is to see the memory retention after modulating the tension in the network. And lastly, we try to reproduce this memory-like behavior or dynamics using a computational model. And I will just show you the summary of it. Here's a schematic of presynaptic neuron and postsynaptic neuron forming a synapse, and they communicate through these vesicles and neurotransmitters. And the membrane potential or voltage of this postsynaptic neuron depends on the firing of presynaptic neuron. If presynaptic neuron is excitatory neuron, then when it fires, the membrane potential of postsynaptic neuron increases. Whereas if presynaptic neuron is inhibitory, then the membrane potential decreases. And it can be described as following mathematical models. So the VI is the membrane potential of postsynaptic neuron, and V synapse is the synaptic input coming from presynaptic neuron when it fires. And we also try to integrate the mechanical tension component into the model. So as shown before, when you pull this or when there is a tension in the network, the number of vesicles increases at the terminal. So one of component of synaptic potential J has this tension term epsilon. So this means when there's a tension, more vesicles, so the synaptic potential increases. And tension also affect this vesicle recovery time constant, meaning when pre-stimulated neuron fires, it releases vesicles and it needs to recover vesicles to prepare for next firing. And when there's a tension in the network, this means that now pre-stimulated neuron can recover vesicle very quickly. And lastly, we also integrated the learning-based model, the history-dependent synaptic potential W, and it has alpha, which is forgetting term. This is a schematic of the neural network consisting of 4,000 excitatory and 1,000 inhibitory neurons. And like the experiment, we shine light in both blue and red region during training, and during recall, we just provide a Q stimulus shining only on red region, and see what activity we can get in neurons under blue region. This is again the small window of the raster plot. This is before any training, just random spikes, spontaneous firings. And this is during training. As you can see, neuron actually, under light, they synchronously firing. And this is a recall phase. When we provide a Q stimulus, you can see neurons under this blue region also synchronously firing. And this is control case where no training is done. You just cue them and you see there's no synchronous firing, but just spontaneous spikes, random firing. And we also measure the synchron index like the experiment over time. So again, no training case like the experimental result show very low synchron index, whereas training case show the synchron index is higher, but decays over time, indicating forgetting again. And the longer duration of training suggests better memory recall ability, synchron index. And one good thing about having this computation model is you can predict beforehand. So what we are actually interested in is memory retention by manipulating mechanical tension. So here we make the neural network we make them relaxed about 20% at this time point, and it shows that synchron index goes down significantly. This means that this network cannot recover quite well. And now we try to make the network contractile, put the tension on it here at this time point to the initial tension level. And surprisingly, it shows the synchron index is actually going back. So it is for us quite interesting to see in actual in vitro experiment whether this neural network can retain this memory after manipulating the tension. So the conclusion, we saw this in vitro neural network has plasticity, therefore, and then we demonstrated the memory-like behaviors, like memory formation and recall and forgetting. And also we manipulated this network behavior by modulating mechanical tension in the neural network. And lastly, we reproduced this memory-like dynamics using computational model. And again, the future plan would be to study actually deeply about memory retention and adaptability after either decrease or increasing the mechanical tension of the neural network. And this is all I have. Yep, any questions you guys have?
So Kyun, how long do these neurons live in your system? When do you start to get the information?
Yeah, my neurons seem to live on up to like 40 days. Yeah, in my case.
Mm-hmm. Yeah. So you saw this firing. Does it take 40 days to mature, or do you get the stable firing even much earlier?
Like to get the stable network burst or stable firing, it's about taking like 10 to like 12 days for me. Yeah. So I can record from 12th day to like 40th day.
Mm-hmm.
Okay.
Any other question? Okay. Let's thank you.
Things that you actually, you know. Oh, I'm supposed to use a mic, sorry. I have a question. This is Nancy for you guys in Zoom. What can you actually train them to do? I mean, what could you do with them?
Like to compute?
Yeah,
But you're-
or even motions or, I don't have a good sense, I think, of what you could actually anticipate, training them to do.
Yeah, the computing part is something that it's, I think, way far away from. What we are trying to do is just trying to see the ability of to like memory, remember, and whether it can.
Yeah, but I mean, if they're remembering something, you're training them to do something, right? And then they do it again, right?
Yeah.
So what's the next thing you could do beyond what you could do right now? Maybe that's one way to ask you. What's like, if you want them to do something, the next
Mhm.
more complicated thing, what could that be?
Good question. Good question. Yeah, go ahead.
So, good question. I need to think about the role.
Yeah, in
Um,
terms of, let's say, if you think, Nancy, in terms of robotics, right? So if you, let's say you train these neurons to fire at 10 at 1 hertz, for example, and that stimulates a muscle and the muscle contracts at 10 hertz. Then, so that's how you train now to walk at 10 hertz frequency, right? And then you start to provide some stimulation. Maybe you just fire once or twice, and then it should continue to walk at that speed, 10 hertz frequency for some time. So that could be one way to think about it. What Kyun is trying to do is to see where if you give a set of image or a one image, can they reproduce that image with partial information, like he's doing 2 blocks as the full information, and then when he uses a single block or single rectangle being shined, then do they remember that it could be part of the 2 block system. So it's still very elementary in the sense of remembering something and executing it for some time. But once these neurons can be trained to do 3 or 4 things, remember 3 or 4 things, then one could imagine combining them to do more complex tasks.
Yeah, so I guess we want to try to think about what interesting thing you could do with that. What if you had a more complex, just more parts to your image? I wonder, would that be any? Yeah, I guess I don't know. Anyways,
The simulation you did, Kyun, you could show the star where you are training them with a star-shaped image, and can it reproduce the star with a small segment of the star? Can it reproduce the star again? So that could be, at least in computationally, that seems to be they're doing, but I don't think he tried with a more complex shape or geometry.
what about merging things? So they learn like, one image and another image, and could you somehow kind of train them so that they would come with some blended version of that?
Yeah. Yeah, there would be something we once thought about it. In simulation wise, it could be doable, but experiment wise, quite difficult. But yeah, that's something that we will keep in mind, merging.
Yeah, that's a very good idea. Yeah, because you could shine multiple images and train with multiple images, right, Kyun?
Yeah. Yep.
Cool. Thanks.
Okay, the next presenter is Saeedur, also from my lab. And Saeedur does mostly experiments. And this is Saeedur's 3rd year going. Go ahead, Saeedur.
Uh, Thank you, Professor. Hello, everyone. I am Saeedur Rahman. So today I'm going to present some of the work that I have been doing as part of the Mind in Vitro project. So let's get started. Okay, so in the human central nervous system, the sensory information, like for example, from retina or the touch sensors that are at the fingertip, those are relayed through multiple pathways through thalamus to different cortical areas for further processing. So after integrating those sensory informations, that can actually lead to different behaviors in mammals or say humans. So now it's obviously going to be very, very difficult to replicate something like this in vitro. But what we can do is we can simplify this greatly. And the model that we have thought about, it has sensory input neurons as in the form of 3D neurospheres, which are derived from stem cells. Those are connected to a cortical circuit, which was formed with primary cortical neurons as a 2D culture. And the external input is given as optical stimulation from outside. Now, when you stimulate the inputs, obviously there's going to be activity in the cortical circuit. And that activity can be read through either electrophysiology, or you can also do calcium imaging to see what the neurons are doing. So that was the in vitro model that we thought of. And this is how we implemented it. So this is an image of an MEA chip, which has underlying electrodes that can actually record the nearby neurons. So the primary neurons, the 2D monolayer that we have, that was seeded first, it's difficult to see it from this image, but you can definitely see the neurospheres, which it seated on top. So on MEA chip, we have a 2D monolayer of neurons, primary neurons, and on top we have 3D neurospheres, which are going to be the input neurons. So what happens after seeding them is that the neurosphere neurons, they form synapses with the primary neurons. Now what we can do is we can provide selective stimulation of this spheroid. Like we can select one spheroid and only stimulate that one and see how the primary neurons, which are close to the electrodes, are responding. Okay, so then we did immunostaining to see the structure better. So here in the green, you can see it's beta-tubulin, which is a marker for the neurons. And in red, there are the channel rhodopsin 2 positive cells, and blue is DAPI actually shows blue, which is the nuclei of all the cells. So in the spheroid, there's like a dense cluster of cells. That's why there is so much blue here, because there are so many cells in the spheroid. And those also have the channel rhodopsin 2, which is the light sensitive ion channel that we use to excite the neurons. And you can see there's a fabric of primary neurons which are underneath the spheroids. So how do we know that this construct actually works? So as I said, what we can do is we can selectively stimulate the spheroids. So that's what we did. So I'm showing the raster plot first. So the raster plot shows the activity of all the detected spikes in all the electrodes. So you can see some two red lines. So the red line is when I start giving the stimulation or the initiation of the light pulse. So you can see I am just showing two light pulses, and you can see there's a response right after I am giving a optical stimulation to the spheroids. But obviously the network has its own spontaneous activity, right? So in between, you can see the network has its own synchronous burst and spikes, but it also responds to the stimulation that we are giving from outside. So here I'm showing the post-stimulus time histogram. So what we did is we stimulated a single spheroid multiple times and see what was the response of it. So this kind of shows the average firing rate after you start the stimulation. So this time is when I'm starting the stimulation, and the green shaded portion shows the duration of the stimulation. So you can see that when you stimulate, there is a rise of activity, or there's more spiking from the neurons, and then it kind of dies down. And obviously, there are some variability. Like one thing you can see from here, the first one actually led to a smaller amount of response from the neurons. And here you can also see like some of the curves, they show like very little activity or response to the stimulation that was given. But on average, you can see like whenever you shine the light or give a stimulation to the spheroids, you see activity from the primary neural network. We also did calcium imaging to kind of like see the neurons getting activated by the stimulation. So stimulation will be difficult to see here. Yeah, so the stimulation will be difficult to see the video, but you can see that this is the spheroid, and you can see the nearby neurons kind of like showing their own activity in response to the stimulation and also the spontaneous activity. So from this data, what we can do is like plot traces for individual cells, identified cells. So and you can see like this is the calcium traces. So whenever there's an activity and the intercellular calcium goes up, you get this like transient signals. So these are when I'm providing the stimulation. So there are spontaneous activity, but also there are evoked activity. And you can see like in response to two of the stimulation, there are like very large activity from the primary neurons. Okay, so now that we know that the construct actually works, that you can place the spheroids and stimulate them to get response from the 2D network, which is your like a cortical substrate or integrator. What can we do with it? Okay, so one thing we did is like we wanted to see if we stimulate different spheroids, is the response the same? Like you have a neural circuit that actually gives the same response to all sort of stimulation. So that's not going to be very useful, right? So we wanted to see that, like if we stimulate with different spheroids, is the response same or there is any difference in the activities of the neurons? So these are the two raster plots for example raster plots for stimulating spheroid 1 and 2, which are denoted here. So if you look at the spikes, so the shaded region is when I'm providing the light. If you look at this spikes, you can see like there is a difference in which neurons are kind of like getting activated earlier and activated later. Like for example, for spheroid 1, there are some isolated spikes, but approximately like electrodes 20 to 40 are kind of like getting activated first. But here, electrodes 40 to 60 is getting activated first. So when we stimulate different electrodes, which neurons are getting sequentially activated, that's different. So there is so the network can actually kind of like distinguish which input it's receiving. So we can see the same population activity in phase space using principal component analysis. So you can see the population activity follows two distinct trajectories if you stimulate different spheroids. But we wanted to understand more, like what's giving rise to this sequential activity. So this actually shows the spiking rate of the different electrodes, like each circle is an electrode and the color indicates spiking rate. So if you look at this for spheroid 1, so if you look at the spiking in or the population activity in for spheroid 1, you can see like there's a cascade of activity that kind of like travels from the top. But if you look at spheroid 2, you will see that that cascade of activity kind of like starts from the right hand side, right? And that kind of correlates with their position. So you can see that the spheroid 1 is on top of the field of the electrodes, and electrode 2 is on the right hand side of the field of electrodes. So whenever we stimulate, these spheroids are likely to be connected to the nearby neurons. So they are passing, whenever you stimulate these spheroids, like they are firing and they are passing along that firing to nearby neurons. And then you have like a propagation of activity. Okay, so there's a science paper from 2020. I think Key before me was also showing some figures from that paper. So in that experiment, they had human subjects and they had electrodes in the brain. So what they did is like they showed the subjects some pairs of words so that they can memorize. So that's the encoding phase, like they're showing them some pairs of words so that they can memorize and recall later. In the retrieval phase, what they did is like they only showed like one word of the pair and asked the subjects to verbally respond to the second word or the paired word. So as Key was showing that you have ripples and associated like bursting activities. So what they did is like they looked at the detected units, the spikes, and then they found that those spikes also kind of like follows a sequence, like we found stimulating the spheroids. The interesting thing is like, so this is the encoding phase, and they have kind of like rearranged all the spiking units based on the average response time. And this is the retrieval phase. And if you look, compare the two, you can see that the encoding and retrieval sequence is very similar. So that means the humans or the subjects are associating a particular word pair with a particular sequence of spiking activity in the brain. So similar to that, we are asking the question like, when we stimulate different spheroids, we see the sequential activities. Now, these sequential activities, can we kind of like use them as a substrate for memory encoding and also retrieval?
Where is this data from?
This data?
Yeah.
This is a science paper from 2020. I can send you the paper. Okay, so what else can we do with the construct that we have with the input spheroid neurons? So we have like a network with defined inputs. So what we did is like we gave like uniformly spaced stimulations for a long time, and we wanted to see like what's the response of the network. Can we get it kind of like can we get it synchronized with the input that we are providing from outside? So you can see that this is the raster plot. So every red line indicates the time that we are starting the stimulation, and we are giving stimulation every 5 seconds, and you can see like every time there is a stimulation, there is like a network-wide response from the network. So we can actually calculate the interval between these two. So these large synchronous activities, we are calling them network bursts, and we can actually calculate the interval between two bursts. So on top left, we have the basal level data, like when there is no stimulation, the network is firing by itself. So you can see like when you are not stimulating them, the network was producing this synchronous burst like with a variable interval. So the interval was between say 2 to about 25 seconds. But when we start this stimulation, you can see it's kind of like getting synchronized with the stimulation that we are providing from outside. You can see like most of the intervals are aligning with the 5-second line. But obviously, sometimes what happens is like you stimulate, like this is not long enough to show that. But sometimes you stimulate, but there is no response, but then it responds spontaneously. So you have some response which is between 5 and 10. And sometimes it skips a stimulation. So the interval then becomes 10 seconds. So you can see like many different points are aligning along the 10-second line. Another thing that was interesting is like, you can see that initially the entrainment was like very high, but as time was progressing, like we stimulated the network for more than 30 minutes. So with time, you can see they are kind of like trying to diverge from the synchrony that was happening at the beginning. So the network was probably kind of like adapting. And another thing that can also happen is like, since we are repeatedly firing the spheroids, it could also be that those spheroid neurons are getting fatigued. Okay. So with the construct, we can also investigate effects of different drugs and or neuromodulators. So as you know, our lab is concerned with mechanical tension in neurons, how it affects the activity of the neurons at synaptic level or at the network level. So I used the drug blebistatin, which Key showed that reduces the tension. And what happens is like if you apply blebistatin, the tension is reduced, the firing also goes down. So this is the spontaneous activity of the cortical neurons, the primary neurons, that if you apply blebistatin, the spiking and the network burst frequency goes down. Then you can do washout and the activity kind of recovers. But what we could additionally do with our construct is like, so one thing is like to see the effect of tension on spontaneous activity, but we could also see what happens to the evoked activity. like with the same construct, we have levastatin that reduces tension and we stimulate through the spheroids. How does that lead to the evoked activity? So initially, this is when there was no drug, the basal level, you can see the response is high. But with blebistatin, the firing goes down and the response also goes down. So with blebistatin, when we stimulate them through the spheroids, the response is low. But if you do the washout and give them some time to recover, then you can see that the evoked activity kind of also recovers. Okay, so moving forward, one thing that I showed is like, can we use the sequential firing of the neurons as substrate for memory encoding and retrieval? What we can also do, I showed like what happens if you stimulate the spheroids separately. But what you can do is like what happens if you provide simultaneous input to multiple spheroids. You can do like context-dependent computation. That means what happens to the response to stimulation to one spheroid when the second spheroid is also simultaneously being stimulated. What we can also do is like in our construct, there was no output. We only had input nodes and a processing unit from primary cortical neurons. We did not have any outputs. But what we can do is like have output units that receives the processed information or firing activity from the primary neurons. What we can also do, maybe form these output units with motor neurons and connect those motor neurons to some muscle tissues. So you can see like you have a sensory input, a cortical processing unit, some motor neurons that are receiving information from the processing unit, and then some muscle tissue so all this can be thought of like an in vitro replication of motor behavior. So that is all I have for today. I'd like to acknowledge my professor, Mr. Tahir Saib, and also my colleague, Joy, who helped with the imaging. Thank you, and I'll be happy to take any questions if you have.
So your main advantage in the system is that you could have one set of optogenetic neurons that you can produce by stem cell differentiation that many labs can do, but you can use them to stimulate all kinds of neurons that are not necessarily optogenetic. Is that right?
Yes, so the input spheroids are formed from differentiating stem cells into cortical neurons. But the substrate neurons, we showed results with cortical neurons, but we also tried hippocampal neurons, and the thing works. So it's not the processing unit or the secondary unit can be from different neurons from different brain regions as well, and primary.
I have a question. So yeah, I don't prepare. Oh, it was a nice talk. So thank you. I don't prepare a sample myself, so I'm sort of like trying to use this sample. But whenever I ask like a biologist to give me a sample, and then whenever I ask them like, can you do this? Can you do that? There's always like a biological difficulties in preparing these samples, right? So like, can you just like maybe give us a little bit of recall in what was like a biological or practical difficulties when you were trying to prepare this like two different heterogeneous samples with the neurospheres and the sheet of samples? Because whenever I ask people to give me maybe try those samples, there's a lot of like time to figure out like how to tune the timing between the cells and how to tune the speed of their growing, all the chemicals that goes into it. Can you give, because like it's in the CS seminar, so maybe can you give us a little bit of ideas like what goes in there?
I would say the main difficulty, like when I was doing this culture, like this heterogeneous one, so we didn't do extensive study of like to characterize like when these connections are getting active. Like when you see the spheroids and if you stimulate the next day, obviously there is not going to be any response, right? Because the synapses haven't formed yet. So the difficulty was like there seems to be a window when this works very well. It's like a few days or over weeks, but then you see that the response is going down. And there can be several reasons for that. So the primary neurons are kind of like very robust, like they have a defined population of glial cells and neuron cells. But with the stem cells that we do, it depends on how well you differentiate. Sometimes you have like a huge population of non-neuron cells, and they kind of like overflow everything and the thing stops to work. So from biological point of view, that's the difficulty. Like, as I found, like you work with the samples and after a time window that passes, then you find that, okay, the response is going down. It's not working as well as it was before. That's the biological difficulty. And there are some other difficulties like from culture point of view, like you have to match them. So what I do is like, I do the primary culture first, and on day 3, I set the spheroids on top. So you have to kind of like synchronize two things, your stem cell culture and also your primary culture, so that both are like, when you are setting the spheroids, you are not too late, and also both are healthy.
Okay, thank you.
Okay, any other question?
I have a quick question.
Yeah, go ahead.
All right. So it seems like your bottom layer of primary neurons is not optogenetic, and then the spheroids are optogenetic, right? And
Yes.
you were just saying that because the spheroids are stem cell derived, they're kind of like hard to determine the composition of it. But why not just use the primary neurons from Q that's optogenetic for the spheroids, I said.
I didn't get your question. Like,
I guess the question is
said-
why using the spheroids? Why not using optogenetic primary neurons as the neurons to be stimulated?
Yeah, that's the question.
so you're saying like we could form this spheroids with primary neuron as well, right?
No, why not use primary neurons that are optogenetic? Why do you have to go a second step of using spheroids? Why not use optogenetic cortical neuron, optogenetic hippocampal neurons directly and
And no spheres. Is that what you're saying?
Yes.
Okay. So I mean, yeah, you can have like a 2D monolayer, and like Q1 was doing, like you stimulate a portion of that, but the idea is like the model. So you have like a cortical circuit and you are stimulating parts of those circuits, right? But if you look at the first figure that I was showing, like in the central nervous system, the sensory information is like a long range. It's coming from a different region. It's a long range signal that's kind of like relayed to another brain regions, which is receiving that input and doing some processing or computation on that. So we could do something similar to what Kiyun was doing, but that wouldn't be like an exact model of that process. So in that case, you are directly manipulating neurons that are part of the network. Did I like, do you have
Sorry,
your...
I meant like I wasn't going for the monolayer. I say like the spheroids you put on top, why not use primary optogenetic?
Yeah.
You can definitely, but
Because wouldn't that be, like you just said, like have a more defined population of neurons in astrocyte?
that would have a more defined, yes, that's true. You can actually form the spheroids with primary neurons, but to look at it from another point of view, like the stem cells, they're kind of like, you can start a culture whenever you want. And right now there's like a huge rush of activity with human neurons derived from induced pluripotent stem cells. So as I said, like we used mouse, but you can also use neurons that are derived from human stem cells and then do different sort of studies. So you can culture the cells, stem cell derived cells separately whenever it's convenient for you. And then you can use that to excite any preparation or not any, but many different preparations of cells, be that primary or any other neurons.
I guess the advantage here is you have just one set of optogenetic neurons, and you can have many different types of neurons that you don't need to be optogenetic. Right? It takes out one requirement away. So if you want to use optogenetic cortical neurons from mice, then you have to have a transgenic mice, right?
Yes. Yeah.
But if you don't want to make a transgenic mice, but you want to study different kinds of neurons, to stimulate them, you just use optogenetic neuron that Saeedur showed, and then that becomes the stimulating means of stimulating all kinds of neurons.
Mhm.
That avoids one step or avoids the extra work of making optogenetic neurons. You just have one optogenetic neurons, and the rest you can study by stimulating them with one source.
Yep.
Okay, thank you.
Thank you.
Okay, if no other question, let's thank both Kiyun and Saeedur and enjoy the nice afternoon outside.